Hi everyone, I'm Millen, and along with Andrew, Salim, and Shale, we'll be talking about transgenic and knockout mice. So, what are transgenic mice? So, transgenic mice are mice that have DNA from another source put into their DNA, and that foreign DNA is put into the nucleus of a fertilized egg of a mice or rat. These mice are used in laboratories to study diseases like cancer and how future medications can affect the disease. So what are knockout mice? So knockout mice are laboratory mice in which one or more genes have been turned off. This is done by deleting portions of the gene's DNA sequence to make it non-functional or by replacing the gene with an altered sequence. So studying knockout mice can provide information about how the knocked out gene normally functions, including the gene's biochemical, developmental, physical, and behavior rules. So let's talk about the history of genetically modified mice or transgenic mice. So in 1974, Beatrice Mintz and Rudolf Janisch created the first genetically modified animal by inserting a DNA virus into an early stage mouse embryo and showing that the inserted genes were present in every cell. But the mice did not pass the transgene to their offspring, and therefore their work on it was limited and impacted. And in 1981, the laboratories of Frank Ruddle, Frank Castanini, and Elizabeth Lacey, and Ralph Brinster, along with Richard Palmater, injected purified DNA into a single-cell mouse embryo, utilizing the techniques developed by Brinster in the 1960s and 70s, showing transmission of the genetic material to subsequent generations for the first time. And during the 1980s, Palmater and Brinster developed and led the field of transgenesis, refining methods and using these techniques to elucidate the activity and function of genes in a way not possible before. So let's talk about why use mice. So mice are preferred species due to their anatomical, physiological, and genetic similarity to humans. And then some advantages to using mice are that they're very small, easy to maintain, they have a short life cycle, and they have an abundance of genetic resources. And they're excellent models to study human diseases with because they're so similar to us. Now let's move on to the big question. Is it ethical to use mice or animal testing for research? And then most people say that emotions are what separate humans from other species. A study conducted in 2006 developed a pain grimace scale for rats and mice and concluded that rats experience pain when they see a familiar rat or mouse suffering. And recently, there was a lot of media coverage on experiments at Japan's Kwansei Gakuin University demonstrating that rats will help other rats in need and even prioritize helping others over receiving a tasty reward. So this experiment placed one rat in a water-filled tank and a second rat on a platform had to figure out how to push open a door to help the drowning rat get out. The researchers observed that the rats quickly learned to open the door to rescue their cage mates. When given the choice between opening one door to save a drowning rat and another door to secure a reward, in which this case it was a chocolate treat, the platform rats helped the distressed rat at first and then shared the treat with the rescued rats. This is very key because it shows that rats have emotions and can help other rats in need. And there are also many alternatives to animal testing. So there's more than 50 alternatives, like computer modeling, previous animal and human DNA, and much more. So when you guys ask a question, I want to know your opinion if you think using rats in research or any animal testing is ethical or not.
So there are three methods that can be used to create transgenic mice. The first method involves infecting a mouse embryo with a retrovirus containing the desired transgene. The second method is to inject the transgene into the pronucleus of a fertilized mouse embryo. And the third method is to inject embryonic stem cells into a mouse embryo to form a chimeric mouse. So on the right, you can see a diagram that summarizes the three steps, but I'll be going into it in a bit more detail. So in the first method, which is labeled as A genetically modified retrovirus is introduced into a mouse embryo, which causes the retrovirus to modify the DNA of the embryonic cells. However, a variety of factors can affect the rate of success for producing transgenic mice in this manner, resulting in generally low success rates. For example, an experiment in 2001 conducted by Nagano et al. Showed that only 4.5% of offspring produced a mouse expressing transgenic traits. Because of its inefficiency, this method is not as commonly used, so I won't be talking about it that much. In the second method, which is labeled as B, it is much more effective than the first method. And in this method, a fertilized mouse embryo is obtained by surgically removing oviducts from euthanized pregnant female mice. Fertilized embryos are isolated from the cumulus mass drained from the extracted oviducts, and the new DNA containing the transgene is injected into the embryos. Under a microscope, a holding pipette will be used to suck and hold onto an embryonic cell, as shown in the picture on the top left. An injection pipette is used to inject foreign DNA into the nucleus of the cell, causing the transgene to randomly integrate into the chromosomes of the zygote. This genetically modified zygotes are then surgically inserted into the oviducts of recipient pseudopregnant female mice. Pseudopregnant mice are essentially female mice that have mated with vasectomized male mice and thus have a copulation plug but are lacking in the male's sperm. As the zygotes develop and grow, all the cells in the mouse will express the transgene since the zygotes carried and copied the transgene during mitosis. Eventually, the transgenic mice are born and each one will hopefully express the desired traits. The DNA in the transgenic mice can be tested through methods such as the southern blot to check for the presence of the transgene. And overall, this method of producing transgenic mice is the most common because it allows for the transgene to be directly inserted into the mouse zygote, which allows all the cells in the mouse to contain the desired traits in their DNA. Although it is widely used, the random insertion of the transgene into chromosomes can vary the degree at which the trait is expressed, which results in some inconsistency. And for the third method, which is labeled as C on the right, the transgenic mice are produced by modification of genes in donor blastocysts. Blastocysts are basically rapidly dividing balls of cells that form a few days after the fertilization of an embryo. And within the blastocysts, embryonic stem cells, or ESLs, can be found. These stem cells are pluripotent, meaning that they can differentiate into any part of an organism. And this quality makes them suitable for insertion into other organisms. The DNA of the ESLs can be modified, such as through the induction of mutations, which can create mutant alleles. The ESLs are extracted from the blastocysts of the donor mice and injected into recipient mice embryos. Typically, this recipient mouse has different observable phenotypes than the donor mouse, such as coat color, to allow for the chimeric transgenic offspring to be easily distinguishable. And you can see a picture in the bottom left of a chimeric mouse with a different coat color. The chimeric mice can be bred with wild-type control mice to selectively breed for the heterozygous mice with the mutant trait.
The key difference between the two is that knockout is targeted. This means that the desired gene is inserted into the specific locus in the target genome via a homologous recombination. By contrast, the transgenic models use random integration. The desired gene could end up anywhere in the host genome. The desired gene might be placed under its own promoter, leading to high levels of expression. This can be good for disease models, leading to an earlier or more robust phenotype. The trade-off is that it's not as faithful a model of the human disease. One possible issue is that the expression pattern might be all wrong. For instance, PRNP in mice is normally expressed more highly in neurons than in glial cells. If a PRNP transgene were overexpressed in glial cells, that would be an ectopic expression and it could lead to a less accurate disease phenotype. From this, we might have less confidence that therapeutic effects in that mouse model would translate directly to humans. In either knockout or transgenic models, the introduced gene can be from the same species or a different species. Just as transgenius doesn't necessarily involve a different species, knockout doesn't necessarily imply the gene is from the same species.
Hello everyone, my name is Salim Safi and I will be continuing the presentation by talking about the applications of transgenic in knockout mice. Transgenic mice are used to study gene function and generate models for human genetic disease. Knockout mice have a certain gene that is inoperative. This is why they are referred to as knockout mice. Knockout mice are used to study gene function in order to validate new drugs and treatments. Utilizing transgenic in knockout mice for research has become crucial in understanding biochemistry on a deeper level. When the gene is disrupted, scientists can examine the effects and the functions of that gene. Transgenic in knockout mice has been useful in numerous fields of research. A research study investigates mice to determine how genes influence the development of alcoholism in humans. These mice in the study were used to investigate alcoholism by way of mediation through chemical pathways in the brain. Research with these mice are crucial in identifying genes and proteins in the brain that play a factor in alcoholism. Transgenic mice are used to study alcoholism by inspecting a gene known to play a role in the disease. Behaviors of the mice are examined to evaluate the role of the gene in alcoholism. Transgenic mice include identifying and isolating a gene of interest for the organism when it is being created. Promoters are stretchers of DNA that are used to turn the gene on. Choosing the correct promoter depends on the location in the brain. Then, fertilized eggs are injected with the transgene. A limitation with the transgenic technique is the inability to connect the transgene back to its original location on the chromosome. If the location on the transgene goes back to its original chromosome as it was identified, it could lead to a potential role in inaccurate analysis. A scientist can mistakenly inject a different part of the chromosome, which can lead to effects that can or could not be due to the transgene. Transgenic mice and their role in research for alcoholism has been used to study developmental processes that can be applied to human diseases. The strategy is useful for identifying candidate genes and effects of alcoholism. The study results show that a low dose of alcohol produced increases in locomotor activity. Also, the transgenic mice played a role in discovering how aggression is affected by alcohol. In creation of the knockout mice, the mutated gene is inserted on the chromosome and in its normal location. Unlike transgenic mice, this eliminates the problem with insertional mutagenesis. The candidate gene is non-functional in these mice. Utilizing knockout mice is great because it does not require multiple lines of mice. This is due to the one copy of the mutated gene being integrated rather than it being inserted randomly. With these types of tests, it is important to keep in mind that many other genes may compensate in response to this experiment. Gene targeting techniques have then become more advanced, allowing for the researchers to maintain control of the study. Knockout mice and their role in alcohol research deals with protein receptors and figuring out how they contribute to alcohol behavior actions. The knockout mice study also analyzed protein kinases. Protein kinases are enzymes that can either activate or deactivate function of proteins. Study has shown that knockout mice demonstrated increased sensitivity to alcohol-induced sedation. Application of transgenic in knockout mice has been useful in discovering more about human diseases. Application of transgenic in knockout mice has been useful since they are similar to humans in terms of genetics, anatomy, and physiology. It is easy to run tests on these mice because it's pretty much an unlimited supply of them. These mice are very easy to manipulate in research by altering their genetics. It has shown to be an effective research method that can lead us to discover how the brain works, development of new vaccines, and many more medical discoveries. I want to thank you for listening to our presentation, and I hope you enjoyed it.